Great. So I'd just like to thank Rick for the invitation to speak here. And I'll be talking a bit differently than some of the other studies today. Specifically, I'll be talking as an ecologist, and given that's my background, and kind of how, as ecologists, we think about infection and immunity, especially when we consider wild systems. So I'll just start really broad, like I usually do, just by stating that we all know, especially in this room, that infectious disease is still a really significant contributor of human mortality and morbidity in humans. And I'm just listing a few of the well-known parasites like HIV, malaria, tuberculosis, and worms that we all know can cause this morbidity and mortality. This is actually just a few of the over 1,400 parasites that have been known to infect humans that are coming from a nice data set by Mark Woolhouse here. And if you just do some quick sums, you can see that co-infection is likely very common in human populations, and this is very similar to what we find in wild populations and is one aspect of what we study. So we've heard a lot about human disease, we've heard a lot about livestock disease, but we also see the same kind of things when we look in wild animals. Often it's a bit more subtle. I mean, we still see cases of animal mortality, especially with emerging infectious diseases, but sometimes it's much harder to see the effects of parasites in natural populations. But we know that they're important and they can structure the dynamics. Here's one example of red grouse that was done in the late 90s. And this is, let's see if I can use this. This is a graph of grouse populations on several moors in the north of England. And as you can see, the population dynamics of these grouse populations are very cyclical, and they have really repeated population crashes where they go to very few individuals. And Pete Hudson and colleagues hypothesized that this might be driven by many factors, one of which might be a strongyloid nematode that infects these grouse. And so they went out to a few moors and figured out when they thought the populations would crash, and they preemptively treated, here in these stars, these populations with an antihelminthic. And as you can see, when they treated, and in some cases they didn't have population crashes at all, in other cases the population crashes were really minimized. So this is a case where it really wasn't obvious that this worm was contributing to these dynamics, but these type of treatment experiments have really shown the impact that they can have. And this is kind of the things I'm interested in. From kind of this knowledge, we have some conventional wisdom that suggests parasites are probably bad for all hosts. But as we know, individuals vary greatly, and I think that variation is going to be important. They can vary in things like parasite burden or the disease caused by a given parasite burden. And importantly, they can vary in their response to treatment and vaccination, and this is something my group is very interested in. And I just want to kind of say that as an ecologist, we think about this variation as a source of excitement. And I know a lot of other people think about the variation as a source of contempt and something that's often controlled in studies. And one reason I think it's interesting can be elaborated in this type of slide. I mean, we know natural populations are variable. They can vary in age, in sex, genotype. If we're thinking about parasites, their exposure to parasites or the immune phenotype that they develop. They can vary just on whether they're infected or whether you sample them across seasons and environments. And they can vary in the infection and co-infection patterns that they have. And all of this variation may be really important if we're thinking about these questions about treatment and vaccination. I think importantly, in a lot of lab studies, we use just a really small focus subset of this variation, and for really good reasons, of course. But I think as an ecologist, we want to kind of break out of that circle and look to see how much will we learn about these processes if we kind of try to incorporate all of the noise that we see in the wild. So kind of the framework that my group uses is like whether we can use this ecological approach to provide a framework for understanding infection and immunity by incorporating the kind of noise that we see in natural systems. So I'm going to talk about a few just snapshots of our work to kind of give you an idea of how we go about doing this. The first one is just asking a kind of basic ecological questions about species interactions, specifically asking whether parasite species interact within a host. And I think importantly, we can think about whether that has interactions for treatment strategies if we're thinking more in an applied setting. We all know that co-infection can be important. I just define co-infection as an interaction between two parasite species. This can be either direct or indirect, and it can be have a negative outcome through competition, through things like shared resources or immune-mediated, or even through direct interference. Or it can actually be facilitation. It can lead to immunosuppression, can lead to one parasite enabling another parasite to be able to infect or grow to higher burdens. And it can also be through things like immune trade-offs or even physical facilitation. But what are the consequences of this co-infection? I think there's pretty good evidence that co-infection can affect things like the severity of disease symptoms, the transmission of infectious stages, and this is really important if we're thinking about transmission in a natural population. It can determine host fitness, and again, what we're really interested in is whether these interactions can actually affect how we think about disease control programs, especially their success in these variable populations. So often we use several different metrics to think about whether parasites are interacting in nature. And most kind of historically, we've used kind of cross-sectional approaches where we can look at a set of animals, see whether they're infected with these different parasites, and then use some statistical approaches to look for associations. More recently, people have used more longitudinal patterns. So in this case, is infection with cowpox virus that make you more or less likely to become infected with something like Bartonella later in life. It's a really nice study by Sandra Telfer in 2010 that showed that using these types of models in a wild vole system really shows how important co-infection can be for future infection. In this case, some parasites increasing the risk, while other parasites decrease the risk of becoming infected with a secondary pathogen later in life. So I think all of these approaches are valid, but as an ecologist, often we go back to using experiments. And so when we're interested in understanding how parasites interact in any natural community, ecologists often use perturbation experiments, and so that's kind of what the approach that we've taken. And we've done this in this wild mouse parasite system. This is the mouse we work with. It's Apodemus sylvaticus, a very common wood mouse in the UK. It has a great number of diverse parasites and pathogens. If we just do a snapshot, we can see that there's all different types of pathogens, from viruses to worms. Coccidia is very common. And in these groups, there's a lot of diversity. On average, each mouse is infected with about 3 parasite species, and so co-infection is quite common. And in fact, about 70% of the mice that we see are co-infected. And you might just think that we chose something that was really, co-infection is really common, but I don't think this is actually that different than most natural systems. So to kind of use these experimental approaches, we go out into populations, and these experiments were done near the University of Liverpool on the Wirral. We can go to kind of just natural populations. They live in woodlands. We live trap the mice, and so we can capture them. We individually tag them with a pit tag so we can follow them throughout their life. And then at each capture, we'll take demographic measures, we'll take fecal samples and blood samples, and in that way we can use diagnostics to put together the parasite community that's infecting that animal. And in this experiment I'll talk about, we use the drug ivermectin as a way to reduce helminth infections in a subset of the mice. So I'll talk about a simple experiment where we had mice that were treated with water, mice that were given a monthly dose of ivermectin at each capture, and then we followed individuals from May to December, taking blood and fecal samples at each capture. And then for reasons that you'll see in the next slide, or in a few slides, we actually went out and did sampling in between these monthly samples for fecal samples. This was about 265 mice and about 600 captures. So if we look at all the parasites that we can detect using our fecal and blood samples, it's really quite diverse range. In this experiment, we're targeting worms, and when I say worms, I really mean H. polygyrus, which Henry nicely introduced earlier. And this is about 90% of the mice that are infected with nematodes are infected with H. poly. It's the most prevalent. It's about 50%. And this 50% is from fecal egg counts. When we actually do dissections, it's much closer to 90 to 100%, depending on when we sample them. And so we were targeting these nematodes with the drug, and we were looking at whether there was interactions between parasites by monitoring other groups. And one group we were looking at is Eimeria. These are coccidia that live in the similar area of the gut. We were also looking at some blood parasites, such as Bartonella, which is a bacteria, and trypanosomes, which are found in about 15% of the mice. So what do we find? Well, I guess not surprisingly, we get a reduction in nematode prevalence, but I will say this is quite a high dose of ivermectin, about 50 times the recommended dose, because the recommended dose doesn't actually remove worms in wild mice. And we only get about a 70% reduction in infectivity in the mice. And as I noted earlier, this reduction is only seen 1 to 3 weeks, so it's a really transient treatment that reduces worm burden. If we look at the kind of dynamics over time, this is just the prevalence of nematodes, but again, it's really H. polygyrus. We gave them a drug treatment here. We see this reduction at 1 to 3 weeks, but I think really importantly, they're all reinfected by 4 weeks. We can treat them again here, we get a secondary reduction, and at 8 weeks they're all reinfected. So it's a really transient effect. There's a really strong force of infection in these natural populations, and the mice get reinfected really fast, and there's no evidence that this ivermectin treatment is giving them any resistance to future infection. So what's interesting is that when we look at other parasites, specifically we're looking at the kind of burden of Eimeria. We see in the same 1 to 3 week time period, we get a significant increase in the kind of oocyst production of concurrent Eimeria infection. And this is suggesting that there might be some type of interaction, a competitive interaction between these parasites in the gut. We see similar patterns if we look at these over time. It's the same graph as here, but again, this is on a log scale, so we do see a several log increase of Eimeria. And again, at 4 weeks, there's no difference. We can treat them again, we see the increase, and at 8 weeks, there's no difference again. So these communities, while they are shifting in response to treatment, it's a very transient shift. As an ecologist, we would call that community very stable to perturbation, in this case, drug treatment. So what's happening? We really clearly don't have the mechanisms known. I mean, one hypothesis might be that these parasites are sharing the same resource or living in the same area. We know that H. polygyrus live in the, oh God, duodenum, duodenum, I always forget what to say wherever I am. So they live there, and they're consuming or living within the epithelial tissue, and this is also where Eimeria are known to, can develop in these epithelial cells. So it might just be that this is competition for the same resources between these two parasites. And we have a little more evidence to suggest this is true. There's 4 different species of Eimeria. E. hungariensis shares the same area as an H. poli infection, and this is where we see the kind of most clear and strong increases after treatment, where if we look downstream at Eimeria apianoides, which lives much further in the small intestine, we see an increase, but it's not significant. So this might be a really local interaction. And again, at this point, we don't have the immune kind of mechanisms to understand if it's immune-mediated, but it's definitely local, which is getting us a little bit further. So if we look across the whole community, we can see that actually parasites do interact, but much fewer than we expected. And so this is just like an effect size diagram. Anything on the left side here is evidence of competition between parasites. Anything on the right is facilitation. And as you can see, the only evidence of interactions we see are really short-term effects with these Eimeria species that are living in a similar area. And none of the trypanosomes or Bartonella species show any effective treatment at the timescale we were able to monitor. So again, this is kind of what we might assume is quite a stable community. I've also done this experiment in the US, and we find similar effects when we treat with ivermectin. We get a reduction in nematodes, and we get a significant increase in coccidia. And this, what I think is interesting about this is a different mouse species, different nematode, and different coccidia species, but we see a very similar result. So there might be something that's quite general about how these different groups are interacting that may be important in this case. The last point I want to make on this before I move on is that I mentioned at the beginning that cross-sectional studies are the common way that we look for interactions. But we did a big statistical analysis, and what we find is that cross-sectional analyses and longitudinal ones, none of them detect the interaction that we see when we use this drug treatment. So we find this negative interaction in both of these mice species. And again, this is the same type of effect size. What we find is that these are all different statistical tests. The tests themselves aren't that important. These are cross-sectional at the end, these are longitudinal. And we actually, using these cross-sectional analysis, usually suggest that these parasites are interacting positively. They actually give us the exact opposite pattern that we see when we do the experiments, which I think is a little troubling because that's the kind of most common way that we look for associations between parasites. And if we use the longitudinal data where we have really good individual repeated measures data, we start to get these negative interactions, but again, it's only 20 or 30% of the time, so it's not really reliable statistics. So I think it's something we kind of have to think about for the future. So the next kind of thing I wanted to briefly talk about was just whether how individuals vary in response to parasitism, and importantly, how this variation may impact health and fitness benefits for treatment. So this is the kind of relationship we see, and this is just H. polygyrus here. This is the egg burden from fecal egg samples, again, because we're monitoring these mice throughout the course of their life, so we can't destructively sample. And this is the survival known in months alive. And this is maybe what we might expect if parasites are bad for a host, that the more, in this case, eggs, or we suggest that means worms, the less long these mice live. And what's exciting, again, is we can use ivermectin to look to see whether this is causal or whether those mice that are infected are just not great mice. But when we give them a dose of ivermectin, we see a significant increase in survival, and that benefit's really only seen in the mice with these heavier burdens. So it is suggested that this worm is having some negative effect on the natural population. What I think is interesting is that this is actually only the pattern we see in female mice. When we look at male mice, we see a really different pattern that's quite interesting. So this is the same set of control mice, but these are males in the wild. And we actually see a nonlinear distribution where the highest survival is seen in mice with intermediate burdens. And I actually don't think the zero burden represents uninfected individuals. I think it represents very low infection where eggs aren't always detectable. And so this pattern is very interesting, and it's been seen in one other system. This is avian malaria and blue tits. And they found a similar nonlinear pattern of annual survival where the birds with these intermediate parasite infection intensities were the most likely to return the next year. So it's an interesting pattern, and I'm kind of wondering what it suggests about parasites and their kind of role in natural populations. But the good thing, again, is we can treat with ivermectin. When we treat with ivermectin, we get a very interesting pattern. So what we actually see is when you treat these intermediate infected mice, they actually get a reduction in about 40% of their longevity. You see a benefit of treatment at really high burdens and at really low burdens, but it's a very different pattern between the males and females. There's something about these, either the infection patterns, the pathology, or the immune response between these sexes that might be driving this pattern, and this is something we're really hoping to look for in the future. So that brings me just to the last few slides. And this is really work in progress, so I won't show much stuff that's done. But we've really focused on the infection side, but I think it'd be really nice to pair this with good measures of the immune response in the wild system, especially if that's going to give us insight into these kind of interesting patterns, especially the kind of importance of natural variation. So we've been working on what we call wild immunology, and the kind of goal, the first goal we had that we started years ago was just to develop meaningful measures for these wild mice. The next goal is then to try to get these measures from serum or fecal samples so we can actually measure things like survival instead of always killing the mice to get spleens or that type of sample. And then the ultimate goal is to match this with our parasitology data. I will say we're just on step one, even after four years, but we've made some progress. So we've got some kind of ELISAs to work, but these are only using polyclonal antibodies and really just with a few of the antibodies, and only one cytokine of all the ones we've tried has worked. And it's just due to the fact that 10 million years between these two mice might mean that there's not enough convergence in the sequence. We've been able to stimulate some cell types, which is giving us a little more data. We've been focusing more on a kind of genomic approach. We now have a genome and a transcriptome, and so we can kind of look through the genome and find ways to measure expression. I think I'll just point out IL-4 here because while we've looked in 4 wild rodents, we've only found it in 1, and it's a non-coding region, so we think that maybe in these wild rodents they don't express IL-4, but that's still a work in progress. So we're hoping to kind of aid this search by, we now have a wood mouse colony here, and this is developed with some colleagues in Roslyn, Bernadette Dudia and Colin Sharp, and I think this will give us a lot more ability to test the mechanisms behind these interactions. We're also looking at diversity of these immune genes across wild rodents when we know the kind of parasites that they get infected with, looking for selection. And we've done some recent work on the wild mice microbiome, which is giving us some interesting insight between interactions with parasites. So I'll just end with a summary. I think there's pretty good evidence that parasites interact in the wild, but these communities are really resistant and resilient to these perturbations, which is somewhat interesting. Response to treatment is really interesting and different for different individuals. I mean, on average, a single dose increases survival by 35%, but as we see, that's very different for males and females and dependent on the burden of infection. I think this is interesting in terms of thinking about improving treatment strategies. I mean, here we show that sometimes it's best not to treat male mice, especially those with intermediate burdens, which I wonder if that's more general than just in this mouse scene. And hopefully the immunological tools will aid us to kind of understand these mechanisms better. And I'll just end with my acknowledgments, especially Andy Fenton, who's a colleague on all of this work.